Hello, myself Gautam from 316. Today's my topic is the structure of muscle and muscle proteins. The structure of muscles is highly organized and designed to enable efficient contraction, which empowers and movement, and muscle structure is organized into a hierarchy of units that work together to produce movement. Muscles are composed of bundles of muscle fibers which contain long thread-like structure called myofibrils. Myofibrils are made up of repeating units called sarcomeres, which are the fundamental contractile units of muscle. Here's a detailed look at muscle structure and the roles of muscle proteins. First one is overall muscle structure. Muscle are composed of bundles of muscle fibers, which are called myocytes, wrapped in a connective tissue. Each muscle has several layers, including three: epimysium, perimysium, and endomysium. Epimysium is the outer layer that surrounds the entire muscle. Perimysium is um, perimysium surrounds the middle layer. It is the bundles of muscle fibers known as fascicles, and endomysium is the thin layer wraps around the individual muscle fibers within each fascicle. Second one is muscle fibers and myofibrils. Muscle fibers, each fiber is a single elongated cell containing multiple nuclei. These fibers are packed within fibrils, the contractile elements of the muscle. Myofibrils. Each myofibril is composed of repeating units called sarcomeres, which are the basic functional units of muscle contraction. These myofibrils run parallel to each other within the muscle fiber. Third one is sarcomere structure. The sarcomere is the fundamental contractile unit with a myofibril. It consists of two types of filamentous arranged in specific pattern. First one is actin, thin filaments, and second one is myosin, thick filaments. Actin filaments are the anchored at the eye line, which marks the boundary between adjacent sarcomere. They extend towards the center of the sarcomere but don't meet in the middle. Myosin filaments are centrally located in the sarcomere and overlap partially with actin. Myosin leads interact with the actin during contraction, pulling the actin filaments inward to shorten the sarcomere. Sarcomeres have striated appearance due to alternating light and dark regions corresponding to the arrangement of actin and myosin. There is a sarcomere here diagram which shows the Z line, M line, Z line, and their bands as I band and A band. There is a zone, H zone, and the fourth one is muscle protein and their function. Muscle proteins play specific roles in muscle contraction and structured stability. First one is myosin. This motor protein makes up the thick filaments. Each myosin molecule has a head that binds to actin, forming cross bridges that pull the actin filaments to the contract the molecule. And actin filaments form the main structure of the thin filaments. Actin has binding sites for myosin, and its movement relative to myosin is what shortens the sarcomere during contraction. Tropomyosin. This regulatory protein binds around actin filaments, blocking myosin binding sites on actin when the muscle is at rest. Troponin. Troponin, it is attached to the tropomyosin and regulates its position. When calcium ions bind to the troponin, it causes tropomyosin to shift, exposing the binding sites on actin and allowing myosin to attach for contraction. Titin. This is large elastic protein, spins from the Z line to the M line and stabilizes the thick filaments, providing elasticity and structure integrity to the sarcomere. Last one is nebulin. Nebulin runs along the actin filaments and stabilizes them, helping to maintain their alignment within the sarcomere. And last one is molecular basis of contraction in muscle. The molecular basis of muscle contraction is rooted in the interactions between the protein filaments and specifically actin and the myosin. Muscle contraction follows the sliding filament theory in which myosin and actin filaments slide past on another to shorten the sarcomere and therefore the muscle itself. Thank you.